Okay, well, good afternoon, everyone. This is Jonathan Maudsley. I am the Science Advisor at the Association of Fish and Wildlife Agencies. I'd like to welcome you to this afternoon's webinar, which is one in a series on fish and wildlife health topics that's co-sponsored by USGS Ecosystems and the Association of Fish and Wildlife Agencies. This afternoon we'll be hearing about dispersal hazards of Pseudogymnoascus destructans by bats and human activity at Hibernacula in summer. It's a webinar presentation with Dr. Ann Ballmann, who's a wildlife disease specialist at the USGS-National Wildlife Health Center. Before we get going with today's webinar, I'd like to take care of some housekeeping details as well as a few announcements. First of all, this webinar is being recorded, and so there will be a recording available on the YouTube channel for the Association of Fish and Wildlife Agencies, which you can find by Googling AFWA, AFWA YouTube channel. And the recording will also be available from myself at the Association of Fish and Wildlife Agencies and through USGS Ecosystems. We want to thank our partners at USGS Ecosystems for making this webinar available, certainly to Dr. Ballmann, to our good friend Dr. Camille Harris, who is out on maternity leave, and Chris Johnson, who is currently on detail filling Dr. Harris's position. So we thank all of them for their support, for the great work that's being done through the National Wildlife Health Center and other parts of USGS on fish and wildlife health topics. So by way of introduction, Dr. Ann Ballmann has been a wildlife disease specialist or field epidemiologist at the USGS-National Wildlife Health Center in Madison, Wisconsin, a federal diagnostics and research laboratory dedicated to assisting state, federal, and tribal management agencies investigating disease in free-ranging North American wildlife. Dr. Ballmann's been at the center since 2008. She earned her doctorate of veterinary medicine at the University of Tennessee, Knoxville, and a PhD in comparative medicine population health at North Carolina State University in Raleigh. She's actively involved in research and multi-agency coordination for bat white nose syndrome and currently serves as the lead for the White Nose Syndrome Diagnostic Working Group, as well as serving on the National Coordination Team for White Nose Syndrome. So at this point, we're going to turn it over to Dr. Ballmann for the presentation, but before that, we will mute the line so that we don't have any extraneous noise, and then I will bring up Dr. Ballmann's line, and we will proceed with the webinar. So hold on here for a technical.
The conference has been muted. The conference has been unmuted.
Okay, Dr. Ballmann.
Yes, thank you very much for inviting me to speak today. I will be presenting the results of a study conducted in the summer of 2012 at 8 caves and mines within the white nose affected region that are used by bats between mid-July to mid-August. And this is describing or investigating dispersal hazards of Pseudogymnoascus destructans by bats and human activity at these sites. Next slide, please. The project was funded in part by U.S. Fish and Wildlife Service as well as U.S. Geological Survey and was conducted in collaboration with state, federal, and NGO partners, including the Indiana Department of Natural Resources, Kentucky Department of Fish and Wildlife Resources, Ohio Department of Natural Resources, Tennessee Wildlife Resources Agency, the Virginia Department of Game and Inland Fisheries, as well as U.S. Forest Service, National Park Service, and the Nature Conservancy, who all assisted with site selection and sample collection. Next slide, please. The presentation will begin with a brief overview of white nose syndrome to provide a background for this study, followed by the research questions that we are interested in investigating, and next I will describe the methods which will focus mostly on site selection and field sampling procedures, followed by the results. And finally, I will discuss the conclusions and implications of the study. Next slide, please. So white nose syndrome is the predominant threat to bat populations in North America, having killed over an estimated 6 million bats since it was first detected in Howe's Caverns in Schoharie County, New York, as indicated by the black arrow on the top map. It was first detected in winter 2006-2007. By spring 2012, prior to the initiation of our study, it had already spread to 19 states and 4 Canadian provinces, and you can see that as indicated by the top map. As of August 2017, depicted in the lower map, the causative fungal agent has been detected on bats in now 33 states and 5 Canadian provinces, with white nose syndrome confirmed in all but 2 of those 33 states, those 2 states being Texas and Mississippi. And this does include the recent 2016 detection of white nose syndrome in Washington State. Next slide, please. White nose syndrome manifests as a whitish fungal growth on the muzzle, wings, and ears of affected bats, causing mass winter mortality or population reduction at the hibernaculum, and results in an array of unusual behaviors such as shifting roost locations to cave entrances and daytime flight observed during periods of sub-freezing temperatures in the winter. White nose thus far has been confirmed in 9 North American species of cave hibernating bats, and exposure to the causative agent has been documented on 6 additional North American bat species, including several migratory bat species. Next slide, please. White nose syndrome is caused by the cold-loving fungus Pseudogymnoascus destructans. Furthermore, I'll be referring to it as PD. It was originally classified as geomyces. Infection trials using PD obtained from sick bats resulted in clinical disease and mortality in naive bats consistent with what was seen in field cases and from which the fungus could be re-isolated. The fungus had not been previously characterized prior to the emergence of white nose syndrome in New York. It subsequently was identified in approximately 15 European countries where it appears to have occurred for many years based on field observations and phylogenetic variation, but has not resulted in the large-scale mortality among bat populations there. Cases of white nose syndrome now have also been reported in China. PD isolates from North America show very little, if any, genetic variation compared to European isolates, which suggests that it has only recently been introduced here and is hypothesized to have come from Europe. Next slide, please. So how does white nose kill? The fungus itself invades the skin of bats during hibernation, giving that characteristic fuzzy white look to the wings, muzzles, and ears. Bat wings play many critical roles in bat physiology during the hibernation period, including maintaining water balance, gas exchange, blood circulation, and thermoregulation. It's natural for bats to arouse from hibernation periodically throughout the winter. White nose, however, causes the bats that are affected to wake up more frequently, and because these arousals are energetically expensive, bats tend to use up their fat reserves before prey is available at the end of winter, and this often then can lead to death from starvation or exposure to the cold while searching for food. Other hypotheses that can suggest death can be a result from white nose syndrome are electrolyte imbalances, similar to what's been described in chytridiomycosis, mortalities of amphibians, dehydration, or heat loss. And finally, damaged wings may also compromise flight for foraging and predator evasion. How it is important to note that not all bats that develop white nose syndrome will die from the disease. Banding studies have demonstrated multi-year survivors in white nose affected areas. Next slide, please. So after the initial detection in New York, the PD had spread in North America in a pattern that is most consistent with a point source introduction of a pathogen. On average, PD has moved approximately 500 kilometers per year, as denoted in the heat map down at the bottom of the slide there. Bat to bat and environment to bat transmission has been demonstrated and are likely the most common routes of transmission. The majority of transmission appears to occur during the hibernation period when bats congregate in an environment conducive to the PD growth and in close proximity to each other, as evidenced by the increasing proportion of visibly affected bats that peak during the late winter and spring period. However, large geographic leaps in PD detection that far exceed any known bat migration distances suggests the possibility for human-assisted movement, and has been proposed as a likely mechanism for its introduction into North America. Next slide, please. So, given that background with the continued spread of white nose syndrome, what was not clear is what is the critical period of when bats most likely move PD into new areas. Surveys of cave sediment over time have demonstrated that PD is present in the Hibernacula when bats with white nose syndrome, or where bats with white nose syndrome are found, and that fungus can persist in these sites even during the summertime in the absence of bats. It's also known that bats continue to use caves and mines during the summer period, and bats therefore could theoretically become exposed to PD when using these contaminated sites during that period and move the fungus to other locations. Summer submissions of bats to date have been fairly limited and mostly opportunistic, and for the samples that we have received here at the center, white nose and PD have not been detected after May each season. Furthermore, we were interested in figuring out what role might humans play in PD dispersal. Next slide, please. So prior to this study, there had been no systematic evaluations of bats that use contaminated sites in the summer to investigate the potential for dispersal risks as bats would vacate these sites and initiate fall swarm activities. So our study addressed the four following questions. Could bats using contaminated sites in the summer harbor viable PD and thus pose a risk for PD movement outside of the winter season? We were interested in what were the appropriate sampling methods for detecting PD in active bats. Could we detect a difference in PD prevalence or relative abundance in the environment when sampled during the summer based upon the different winter mortality effects seen at these Hibernacula sites? And finally, what was the potential for human-assisted movement of PD associated with summer trapping or caving activity in the white nose affected region? Next slide, please. So to address these questions, 8 Hibernacula with documented summer bat activity were selected from within the white nose affected area based on evidence of summer use by bats and the relative ease of trapping at a single entrance location. The small inset map identifies the 5 states where the 8 Hibernacula were located, and then the larger map denotes the county level resolution of where one site per county was included in the study. 6 sites were known to be contaminated with PD, and those are indicated on the larger map in red. And these had been known to be contaminated as early as winter 2010-2011, so anywhere between 1 to 2 years prior to our study. And the 2 sites in blue noted on that map were noted to be visibly free of white nose as of spring 2012, before we started our study. Surveys were conducted in mid to late summer prior to fall swarm to assess the prevalence of viable PD on bats occupying contaminated sites that would then likely leave these sites and transport or move to other locations during fall swarm. Next slide, please. So the next slide should show a table. It compares various characteristics among the 8 study sites. The county and state of origin of each site and the site type and access are identified in the first 2 columns. And the slide hasn't changed yet, but if you switch over to the next slide. White nose status in the table indicates the status of the site at the end of spring 2012 and the year white nose or PD was first detected. Winter bat populations were classified as either stable or reduced based upon the winter 2012-2013 winter counts, divided by the average of 2 to 5 years of pre-white nose winter census data, which would include the year of white nose detection. Species hibernating at each site during the winter appear in the last column of the table. And for clarification, the green color coding in the table corresponds to negative control sites where white nose was not visibly observed at the sites at the time of sampling, or sorry, the previous winter had not previously been observed. The blue indicates the PD contaminated sites that had maintained stable winter bat populations, and then the orange indicates PD contaminated sites where there was at least a 25% decline in winter bat counts since white nose detection. Okay, next slide, please. Bats targeted for sampling were those collected inside or trapped exiting the site at evening emergence for no more than 3 trapping events. Power analyses determined that the minimum sample size required to detect at least 1 positive bat when we estimated a low summer prevalence between 2 to 5% would be between 45 and 115 individuals per site. Harp traps, mist nets, and or hand capture techniques were used depending on the site to achieve the targeted sample size. And although not sampled for PD, bats captured on outside of cave entrances were also recorded to assess the overall summer bat activity at the study sites. Trapping equipment was provided by the local collaborators and decontaminated in accordance to the current U.S. Fish and Wildlife Service guidelines and state requirements at the time. Furthermore, to avoid cross-contamination, new disposable gloves were used for each bat and environmental sample that was collected. Bats were held in individual bags until processing, and guano was collected opportunistically from the bat holding bag. Basic demographic data were collected from all sampled bats using standard techniques. And what was collected is identified there in this slide. Wings were assessed for level of wing damage and inspected under long-wave UV light for fluorescence, which is suggestive of white nose syndrome. And finally, a wing swab consisting of the entire dorsal surface of one wing was collected from each bat for real-time PCR targeting the highly specific and sensitive IGS region of PD, which has been described previously by Moeller et al. In 2013. And fungal cultures from wing swabs were completed from a small subset of bats, totaling 56 out of those sampled. All sampled bats were marked with temporary paint on the dorsal fur, as well as wing banded in many cases, depending on the preferences of the collaborating agency, and this was to prevent repeat sampling on subsequent trap nights. Next slide, please. So in addition, paired environmental samples, both swabs and floor sediment of equivalent surface area were collected from 8 locations inside each study site for a total of 16 environmental samples per site. Locations roughly corresponded to areas where bats were known to roost during the winter time, or areas where bats were observed roosting at the time of collection, or there was at least evidence of fresh feces suggesting recent roosting activity. PCR was performed on these samples to estimate the relative abundance of PD and compare the level of contamination among sites. And finally, swabs were collected at the end of each trapping session from various equipment in direct or in indirect contact with bats or cave environments to assess for possible PD contamination. And again, real-time IGS PCR and fungal cultures were performed on a total of 53 samples. Next slide, please. So moving on to results, this table shows the late summer bat activity and trapping methods used at the 8 study sites. A total of 617 bats representing 7 different species were captured from inside the 8 study sites that were sampled. And then there was another 234 bats that were recorded at the outside of these study site entrances. We were able to obtain at least the minimum sample size or targeted sample size from all 8 sites. Next slide, please. So the sites with stable total winter bat populations tended to have a higher density of summer bats, and this is indicated by the red box in the capture effort column. Next slide, please. And recapture rates were extremely low among the 7 study sites where entrance trapping was used. This suggested that frequent movement of bats using underground roosts in white nose affected areas during the summertime, it suggested that there was frequent movement of bats using underground roosts in the white nose affected area during summertime, and in theory could contribute to the movement of PD on the landscape if exposed to PD. Next slide, please. These pie charts illustrate the summer bat diversity sampled from inside each study site. They are grouped here in this figure in rows based on site classification, where the top indicates where a PD contaminated site where the winter bat population was considered stable. The middle row are negative control sites, so no white nose had been previously detected at these sites, and again the winter populations were considered stable. And finally the bottom row are PD contaminated sites that showed at least a 25% decline in winter population. The common names of the species appear in the key on the right and correspond to the 4-letter genus species code labeled on the pie chart itself. The number of bat species detected in summer was either the same or less than those observed in winter historically. However, little brown bats and tricolored bats, 2 of the species that are most commonly diagnosed with white nose syndrome in the wintertime, were detected at all sites during the summer in our project. And there were no migratory bat species captured from inside these sites during the summertime, although we did have an eastern red bat presence observed at the entrance of one of our study sites in Montgomery County, Tennessee. It was in very low levels, only 2%. Next slide, please. So males outnumbered females captured at all the study sites, including the site at Trigg County, Kentucky, which contained an active gray bat maternity colony. Overall, males significantly outnumbered females by more than 4 to 1, and this agrees with findings of previous trap surveys conducted at or near caves and mines in the summertime. Males and non-reproductive female bats have a lower energy requirement than reproductive females, and can tolerate roosting at lower roosting temperatures and tolerate more frequent torpor bouts in the summer to conserve energy. Next slide, please. So to summarize what we found in the laboratory, PD was detected at 7 of 8 study sites during the late summer 2012. 40 bats from 6 of 8 study sites tested positive for PD, including one of the negative control sites that's indicated by the yellow arrow in the table. Here, a single juvenile male tricolored bat tested PCR positive on wing skin, representing the first detection of fungus at this particular location. All environmental samples collected at this site in August 2012 tested negative for PD. However, white nose was later confirmed at both negative control sites the following winter. Next slide, please. So PD detection, you'll note, was greater in guano and soil than was in wing or wall swabs, respectively. Only 4 of 617 bats, or less than 1% of the bats sampled using wing skin, tested positive for PD, while guano from 37 of 223 bats that cooperated and provided us with guano tested positive for PD during that same period. Guano accounted for 93%, or 37 out of 40 of the PD detections on bats that were sampled during the summertime. And this is thought to be due to grooming activity and ingestion of PD spores off the skin surface of the bat during periods of activity. Bats of both sexes and both adults and juveniles had evidence of exposure to PD. And viable PD was recovered from the wing swab collected from a post-lactating little brown bat female that was sampled in mid-August. None of the bats sampled had any visible evidence of white nose syndrome infection during the sampling period. And nearly all bats, roughly 97% nearly all bats examined had no to minimal evidence of wing damage despite their use of these contaminated sites. 3 bats that had some suspicious UV fluorescence all tested negative for PD by PCR and histopathology. And species testing positive for PD, whether it was by wing or by guano in the summertime, included little brown bats, Indiana bats, gray bats, eastern small-footed bats, and tricolored bats. Not surprisingly, PD was detected from environmental samples at all 6 study sites where white nose had been previously confirmed in their winter bat populations, and not from the environmental samples at the 2 negative control sites sampled at the time. Next slide, please. So not only was PD more often detected in sediment samples, it was also found in higher relative abundance, which is represented by the lowered CT values noted in the top graph on this slide. It was found sediment samples for PD was found in sediment samples at a higher relative abundance than from swabs of walls or ceiling substrates that were taken from the equivalently sized area. On average, PCR amplification of PD occurred in 10 fewer cycles in sediment samples compared to the wall-based samples. There was no significant difference in relative abundance of PD detected from environmental samples that was associated with white nose population level effects, however. In that top graph, it may be a little bit hard to read, but the x-axis identifies soil to the left and results of soil to the left and wall swabs on the right. And the darker or black bars are results from sites where they had PD-contaminated sites that had stable winter bat populations versus the lighter gray, which were PD-contaminated sites with reduced winter populations. On the bottom graph, controlling for species differences between sites, summer bats at sites with reduced winter counts were less likely to test positive for PD than bats tested at sites with stable winter counts, which was not what we had expected to find. This appeared to be due mostly to the high prevalence of PD encountered at one particular first-year white nose positive site, which accounted for 70% of all the PD detections in this bats that were sampled in this study. Next slide, please. So as for the potential for human-assisted movement of PD, this table on the left shows the types of items that were sampled for PD contamination. The items are categorized as trapping equipment, of which we had 29 total samples on the top. Next was clothing or personnel, which is in the middle, where we had a total of 18 different samples collected. And finally, processing equipment at the bottom, where we had 6 different items sampled in that category. All swabs were collected at the end of each trap night or site entry. The number of gear samples collected per site ranged anywhere between 5 to 13 and would always include trapping equipment when applicable. Mist nets were swabbed over the entire surface area with a single polyester tip swabbed, and different parts of the harp traps separating between the vertical filaments and the catch bag were swabbed separately to assess areas of possible PD contamination. We detected PD genetic material on harp trap catch bags as well as harp trap strings, or the filaments, and as well as backpack used at 3 of the 6 different study sites where white nose had been previously confirmed. Equivocal results indicate that amplification of PD material was observed, but was below the threshold of sensitivity set for the assay. Although no PD was detected from mist nets in this case, the sample size was much smaller compared to that of the harp trap sampling. Viable PD spores were not demonstrated in culture, likely due to low levels of PD material that were detected. However, because viable PD was demonstrated from at least 1 bat during the study, it remains theoretically possible that for bats to contaminate trap equipment with viable PD. Next slide, please. So returning to our study questions, what can we conclude? So when looking at can bats contaminate sites in the summer, or bats using contaminated sites in the summer harbor viable PD and thus pose a risk for PD movement outside of the winter season, our study demonstrated that bats that do occupy these hibernacula in the summertime face ongoing risks of exposure to the pathogen. Although exposure to PD does not appear to result in disease during the summer months, our results demonstrate that bats can carry the viable fungus on their skin and thus facilitate bat-mediated movement of the pathogen during mid to late summer. It's particularly evidenced by the detection of the bat at the negative control site, which was the first detection. Next slide, please. What appears to be the appropriate sampling methods for detecting PD in active bats? We found that to optimize PD detection in non-hibernal bats, sampling should include the collection of both fresh guano and skin swab samples from individuals. And why do we recommend sampling both when there was such a clear indication that guano resulted in the majority of PD detections in this study? We do recommend both because not all bats will cooperate and provide fresh guano pellets for testing, and the fact that PD wasn't always detected in the guano of an individual bat with a positive skin swab sample. So by using both sampling methods, it increases your chances of detecting PD exposure if it's there. And when environmental samples are collected for PD surveillance in the absence of bats, or when disturbance is inappropriate, the sampling should prioritize sediment samples or soil samples from the floor or rock shelves collected underneath areas where bats roost rather than vertical wall or ceiling substrates. And we hypothesize this to be true due to gravity acting on PD spores that may be shed from the skin or through the guano of bats. Did we detect difference, or did PD prevalence on bats or relative abundance in the environment during summer differ between sites experiencing different winter mortality? The answer is both yes and no. As far as PD prevalence on bats, it was higher at sites with stable winter bat populations, but this was likely due to differences in species composition among the sites. As far as abundance in the environment, the answer is no. The effects of white-nosed winter mortality did not influence the prevalence or abundance of PD detected in the environment among these sites. Because PD is known to persist and grow inside Hibernacula in the absence of bats, its relative abundance in the environment may be multifactorial and not so much, it includes something in addition to certain abiotic factors may be involved in addition to simply white-nosed population level effects. And finally, the fourth question, is there a risk for human-assisted movement of PD associated with caving activity or trapping activity in the white-nose affected area? Yes, the study demonstrated that equipment used at Hibernacula known to harbor bats with white-nose syndrome in winter can become contaminated with PD during the summertime. PD was identified both on equipment that was used exclusively outside the Hibernacula entrances and on equipment that was carried inside the Hibernacula. And this all occurred during periods of low PD prevalence on bats. Although the viability of the fungal pathogen was not demonstrated by our study, this was not unexpected given the high PCR CT values that were obtained from the PD positive samples. So next slide, please. So what are the main implications of this study? With regards to decontamination, bats and people entering underground sites in white-nose affected areas during summer risk exposure to viable PD. Therefore, routine decontamination of equipment, including mist nets and harp traps, following use at environmental reservoirs during the summer, including in the summer months, is warranted, particularly in areas where the risk for geographic expansion of PD is greatest. Results from this study also inform surveillance efforts by identifying the best practices for sampling. We demonstrated that to optimize potential for detection, surveillance for PD on active bats should target fresh guano in addition to wing skin. We recommend that collecting both samples from bats that are trapped or recovered from the landscape during these non-hibernal periods. Although our findings demonstrate that guano is a more reliable source of PD detection in active bats, we remain uncertain if this will remain the case when applied to pooled guano samples, particularly those collected from above-ground environments. So further study is underway to determine the reliability of guano as a non-invasive sampling technique for the detection of PD in new areas that would not require handling bats. Furthermore, we demonstrated that sediment collection should be prioritized over wall swabs at Hibernaculum to increase the likelihood of PD detection. With regards to the timing of surveillance activities, we demonstrated that targeting bats that use underground roosts during the summer can be useful for detecting PD and presents an alternative to conducting surveillance during the winter season when Hibernacula may not be accessible. One caveat being, however, though, that larger sample sizes per site may be required if sampling in the summertime. And finally, more study is needed to characterize the role of guano, or to characterize the role guano may serve to disperse PD during the summer months. If PD can be demonstrated to survive GI transit after ingestion, bat movements between underground sites during summer and fall periods could help explain how PD moves on the landscape. Next slide, please. So I just wanted to thank the many volunteers and collaborators that assisted with sample collection at all these sites. It was quite an endeavor, and we appreciated everybody's assistance to see the success of our project. And with that, the next slide is time for questions.
Great. Thank you very much, Dr. Ballmann, for that excellent presentation. We really appreciate sharing this information with the AFWA audiences. And I think some of you have probably seen the little chat box in the corner. We've been soliciting questions through that chat box. We already have a couple, which I will start with for the Q&A, and then as time permits, we will go ahead and move on to a more traditional Q&A with opening up the lines and then having people ask questions. But for now, we'll start with a couple of questions that have already come in the chat box for Dr. Ballmann. The first question is from Nathaniel West. If a bat survives, is it immune afterwards?
We do not have information on the survivability or through immune function on bats. Some studies have looked at I guess the antibodies that may be produced after exposure to PD and have found that they do not mount a significant antibody reaction to that. But there is obviously evidence of survivors that can maintain, but the mechanism for that survival is still under investigation.
Great, thank you. Question from Amy Hamasfar. Will the results of this study change the talking points and public educational messages that are being provided by the white nose syndrome working groups about white nose syndrome transmission?
I don't believe it's going to change what's currently out there. I think the evidence that we have supports obviously the need for year-round decontamination, again, particularly at those areas where the risk of PD movement is the greatest, so kind of at the leading edge region. So I don't think that will change. As far as making sure that people who go to these sites are aware that one, PD can exist in the environment in the absence of bats is unchanged. The fact that bats may be harboring PD during the summertime is a bit of new information, but again, it's not going to change the public messaging.
Great, thank you. Question from Robert Shore. For those interested in collecting fresh guano samples from captured bats, what is the preservation collection methods you recommend, especially if you're planning to submit those samples to a testing lab later on?
The holding or sample holding methods that we used was simply to place individual guano pellets from bats into a sterile tube with a desiccant bead in it to prevent further fungal overgrowth that might occur when there's large amounts of feces present, and that seemed to be sufficient. Once we did that, we then placed it into -20 storage for a short period before we could get back to our lab here in Madison, which we then stored it at -80. So if it's going to be prolonged storage, I would still recommend, even with a desiccant bead, storing it at a -80 if possible.
Great, thank you. Question from Lori Hanuska Brown. Soil can be hard to find at some sites. Do you think gravel, sand, and rock provide the same medium for PD persistence?
I know that it's without sediment samples in the sample, there's nothing to it. You can't really extract DNA from the inorganic material, the rocks and things like that. And I think some of the work, when we're, I guess, counseling people to collect samples, we do ask that they try to avoid, those gravel and rocks and things and such. In the absence of sediment samples, the next best thing that would be to collect would be, a swab of the floor or the shelf of the cave underneath or areas where bats roost, as opposed to trying to physically collect the sediment.
Great, thank you. Question from Rita Dixon. Is it worthwhile to collect guano and wing swabs during mist netting on the landscape, or only during summer of bats at the known Hibernacula?
I would say targeting bats that would use underground roost sites would be the priority. What we have seen thus far from the surveillance samples that we have received through the surveillance project that we're doing has not been very productive in detecting PD on bats that were just trapped off of random areas on the landscape itself. But I would certainly, if that's an option of trapping bats at a underground site that they use during the during the summertime, then those would be the priority bats to target in the summertime. So a more specific targeted location for sampling.
Thank you. Follow-up question from Lori Hanuska Brown. What's up with finding PD on a backpack but not on clothing? Any thoughts as to how that might have happened?
It's just the random draw of where you might have swabbed to pick that up. I agree, looking at some of I think one of the pictures I included in the presentation where we're just covered all of our backsides in mud. It's surprising that, I'm guessing PD would have been there somewhere, but we just didn't collect it at the location where we swabbed. Whereas, backpacks that get that get carried into some of those sites and set down on the ground as well get covered in some mud. So again, just maybe random luck of the draw of finding it where we swabbed.
Thank you. Question from David Riggs. The equipment contamination slide contained the phrase 53 fungal samples all tested negative for PD. Could you please clarify if or what equipment tested positive?
So the positive detections on the equipment were based upon PCR detections only. For all of those, we collected 53 swab samples off of the gear, and we ran PCR as well as fungal cultures on all 53 of those samples. None of the fungal cultures from any of those items came back positive or with a viable PD detected, but there were, again, the 3 items that did have PD detected by PCR alone.
Great, thank you. And that concludes our questions that we've received through the chat box. What I'm going to do now, since we do have some time remaining in our hour, and being respectful of people's time, we do have time for some more traditional questions and answers with Dr. Ballmann. At this point, I'm going to unmute the lines.
The conference has been unmuted.
Once the lines are unmuted, if you have a question for Dr. Ballmann, if you could please state your name and your agency or organization or affiliation, and then your question. We'll work through questions that way. So at this point, the line is unmuted, so if there are other questions for Dr. Ballmann, please feel free to come forward at this point. Thank you.
And this is Rita Dixon. I have a kind of a follow-up question. Am I still connected?
You're on, yes.
I hear you.
Oh, I
You're live.
thought my phone dropped the call. Sorry about that. So here's an example, Anne. We have some sites that have not been documented as Hibernacula, like some of the abandoned mines that have had documented bat use during the summer. So from your perspective and the results of your study, do you think it would be worthwhile to maybe net or harp trap at some of those and collect guano and wing swabs, even though we have no evidence that they would use those during the winter?
I think that it may be worthwhile in the fact that you can use the bats that might use these sites in the summertime as sentinels for sampling the site. So this study did not look at specifically I guess let me start over. So there is evidence out there from other studies that demonstrate that for early PD detection, sampling bats gives you an earlier point of detection when PD is introduced versus sampling environmental samples alone. So in the case that you described here, if you have sites that you don't know what the status is of the PD status is at the time, but you know that there appears to be some bats, and I would guess I would have to be a fair number of bats that are observed to be using that site or visiting that site in the summer, you could use those bats as a sentinel for sampling that underground roost site for PD. Again, thinking that we might be more likely to detect early detection of PD introduction into those areas by sampling the bats as opposed to relying solely on environmental sampling at that given site. Does that make sense?
Yes, thank you.
Other questions?
Anne, this is Lori. I guess maybe it's not that pertinent to management, but if someone trying to answer that question you hinted at, is the fungus, I don't know, transmissible after it goes through the GI tract?
Oh, is it transmissible? So we've done preliminary work that suggests that yes, in fact, PD can survive GI transit and remain viable. So we have been able to culture it from guano that's been taken directly out of the colon of those bats. What we're doing a further study at this point to just see how common that might be. But the thought would be, again, if PD survives GI transit and can remain infectious, presumably, whenever the bats ingest it and move and defecate in the new areas, they could potentially seed a new environment if that environment is conducive for PD growth.
Thanks.
Any other questions?
Hi, Anne. It's Scott McBurney from the Canadian Wildlife Health Cooperative. I just have a question about Figure 2C, where the walls were tested, and the CT values are quite high, all of them above 40. So would you really consider those to be positive samples at all, as opposed to just being in a contaminated environment?
So our cutoff value was 40 for our assay detection limit. The way that the data was analyzed, it was modeled to allow for CT values that might appear higher than that 40 level cutoff. However, so the CT values, and you're specifically talking about gear. I'm sorry, Scott, is that what you were asking about, or?
I was asking about whether or not you would consider the walls of the cave actually positive for PD based on those very high CT values, or whether that would just be reflective of basically handling things in a contaminated environment. I mean, my understanding is that would represent such a, probably only a proportion of a spore of PD being that high.
Right. Certainly, there were some that were kind of in that zone that we are now potentially considering to be potential for PD contamination. But I guess what I was trying to show with that graph is that the sediment samples had distinctly lower CT values, and so that was one of the reasons we detected it more readily off of the sediment samples and at higher relative abundance than what we saw off the wall. In our experience, we have not detected PD off of environmental samples without also getting positive detections on bats at the same time. I don't know if that's always been the case with other groups that have been sampling.
Thank you very much. And that graph does very much highlight exactly what you're saying, that those soil samples are a much better place to place your bet than trying to get anything off the wall. Thank you.
Other questions?
Hi, this is Rita again. Just a follow-up, and if we do want to collect guano, because we already have some of the little vials from you for winter sampling, so should we just use those, or are there other ways we should be collecting it?
If you're collecting guano from individual bats, which is what we would be recommending, if you're trapping bats and getting concurrent wing swab and guano samples, then you can just use those vials with the desiccant beads. Work we're doing this summer is looking at the utility or possibility of collecting pooled guano samples at some of these sites and using that to screen for the presence of PD, but we don't have those. Because it's still in the investigative phase, we're still trying to work out what might be the best or most appropriate sampling method for that.
Great, thank you. Are there other questions for Dr. Ballmann? If not, I'd like to thank everyone for joining us this afternoon for this very informative and important webinar on this very significant topic. I'd like to especially thank Dr. Ann Ballmann from the National Wildlife Health Research Center at the U.S. Geological Survey. I'd like to thank our friends and partners at USGS-Ecosystems for co-sponsoring today's webinar with us, particularly Dr. Camille Harris, who is on maternity leave, Chris Johnson, who is on detail filling Camille's position as the National Fish and Wildlife Health Coordinator at USGS Ecosystems. I'd like to thank all of you for joining us today for today's webinar. As a reminder, our webinar has been recorded, and the recording will be available at the Association of Fish and Wildlife Agencies YouTube channel, which you can find by Googling AFWA, AFWA YouTube channel. We'll also make the recording available through USGS Ecosystems. And on the webinar flyer, there is a link to the forthcoming paper on Dr. Ballmann's research, which was presented in the webinar today. So thank you all very much for joining us. Thank you, Dr. Ballmann, and hope you all have a wonderful afternoon. This concludes today's webinar. Thank you.